Hello, everyone, and welcome to part one of the lecture on chapters 11 and 13, bits and pieces from both of those. We're going to be covering general characteristics of bacteria and some general information about viruses and prions, which are subviral particles. They're totally bizarre. The virus and the prions, what they have in common is that they're acellular, where all the other forms of life are cellular in nature. We've got some things that are acellular, and we'll talk about those in turn. But let's start off with the bacteria. Some of this is going to be review. Some of the typical bacterial morphologies are things like the coccus shape, those round balls, and the bacillus, the rods. There can be some things that are kind of in between those two shapes. Coccobacillus, it's referred to. Vibrio, kind of a angled shape. A few that I have not mentioned to you before, spirillum. These are stiff, and the spirochetes, those are more flexible. They're kind of spirally in nature. And then there's some that just, they're just different shapes. The term for this is pleomorphic. And then another characteristic that we keep track of is whether these species can form what are referred to as endospores. I'm not going to go into a huge amount of detail about what that is, other than these are, I don't know how to describe it other than, the environmental conditions are getting tough, and these certain bacteria have the capacity to kind of hunker down for these bad conditions that are coming up. And they can survive ridiculous lengths of time with no water and extreme temperatures and extreme changes in pH. They're really, really hardy things, which is great for them, but not so great for us when we're trying to fight against them. So some groups of bacteria, some of the gram-positive bacteria, some bacillus, and a group known as the Clostridium group. The Clostridium, man, these. You might not know the names yet, but if you've ever heard of tetanus and people needing tetanus shots, it's because there's a species called Clostridium tetani that lives in the soil, and it can survive ridiculous lengths of time. If you've ever heard of somebody like, did you step on a rusty nail? It's not the rust that's the problem. It's these endospores of Clostridium that can survive in these really inhospitable conditions and can cause infection in us. It's a defensive strategy for the bacterium. It allows them to survive, but it makes it really hard to clean certain things. There's a Clostridium difficile. You may know of it as C. diff. It causes terrible diarrhea. And many of our hospitalized patients end up getting it because it's so hard to get rid of. I mean, just on light switches and, you can go over it with bleach and you can try to clean it off, but these things can kind of survive that. And then as soon as they get into the gut, party, they can start reproducing. It just really, really hard to get rid of. So these are of concern to food processors. I mean, commercial food kitchens have to be concerned about this because it's really hard to clean out of equipment. It's hard to clean off surfaces. I'm trying to think. So Clostridium botulinum. So if you've ever heard of somebody getting botulism, it's because of one of these species that can really survive these very, very bad conditions. And so, you boil the things. If you've ever heard of the process of canning, it used to be an old-timey thing that people would like harvest their foods at home and then boil them and boil the jars and then put these lids on them, and they're supposed to last for months and months. But if they didn't boil it quite long enough, the Clostridium botulinum could get in there and could cause problems months later when people ate that food. Healthcare professionals, it's Clostridium difficile that's the biggest problem. And yeah, these things, I'm going to show you pictures. It doesn't matter. The point is that they can survive for really, really inhospitable conditions. So it's very hard to get rid of them. The method of reproduction for most bacteria is something called binary fission. It's a little less complicated than mitosis. Um, so yeah, binary fission is the most common. A parental cell splits into two daughter cells. So what has in common with mitosis, cell division, is the DNA does have to get copied, but then the cell just splits into these two bits. There's no nucleus to worry about. Remember, the nuclear envelope has to dissolve in the eukaryotic cells. Doesn't have to do that because this is bacteria and there's no nuclear envelope to worry about. And then the arrangement of the cells is used still as we describe them, and the arrangement of the cells has a lot to do with how the binary fission is occurring, like what plane of the cell is dividing, and I'll show you what I mean. And then whether those cells like stay touching each other or whether they split apart completely. So planes in which the cell divides, and then the separation, when they come physically apart from each other, like so. So if the plane of division is like this, and the two cells stay together, these are diplococci. We can have streptococcal arrangements where the cells continue dividing and they don't separate from each other. But then if you separate in two different planes, I'm going into way more detail than you need. You get the idea. Split them this way, split them that way. You might get tetrads, or you might get sarcini, or the grape arrangement looking like a bunch of grapes. That's the staphylo arrangement. And that can happen either with the coccus shape of bacterium, or it could be the bacillus shape. You can have streptobacillus. Oh, palisades, I've never mentioned these to you before. So the cells just stick together. And could be just a couple of them stick together in this V shape. There's some matching things or identifying things in the homework this week. You'll figure it out. Oh, okay. Now I understand what this was. I just wanted the pictures to be bigger so you could see it a little bit more easily. So this palisades arrangement is when, I don't know, I think of them as they look like logs kind of near each other, kind of holding on one end. All right, nowadays, now so that was, we based classifications on that kind of thing. Like, what does it look like? How does it stain? The gram positive and gram negative. It turns out that some of those ways of classifying things based on like what the bacterium looked like actually did have some relationship based on like evolutionary relationships of these different groups. So our modern prokaryotic classifications depend on the sequences of nucleotides. So let me see if I can give you an analogy. So like if you study birds and you study the evolution of birds, you do things like, the shape of the wing, the bones that are shaped quite like this, and then the shape of the beak is like this. So you look at characteristics that they all have in common and see which ones are the same and which ones are different. And then you create these trees based on what you imagine are the evolutionary relationships. All right, perfect. Except you're looking at bacteria, they don't have wings, they don't have beaks. What are you going to use? And so one of the things that these organisms, these cellular organisms all have in common, all of us have in common, are ribosomes. We all use ribosomes to do that translation step of the central dogma of biology. Well, these ribosomes are nucleotides, ribosomal nucleotides in orders, the A's, U's, G's, and C's. And so that is the structure. And so instead of looking at the shapes of the bones, we're looking at what's the nucleotide sequence of these ribosomal of these RNA sequences that make up the ribosomes. Huh, brilliant. Well, when you look at those sequences, you get three main domains of life. The domain bacteria is where all the bacteria end up. The domain eukarya, that's where all the eukaryotic organisms, that is all the cellular organisms that have that separate nucleus where the DNA is all in that nucleus. And then there's this group that nobody really talks about, the domain archaea. And these are just some of the bizarro single-celled organisms that kind of popped up. We all realized they existed when we started looking at these rRNA sequences, ribosomal RNA sequences. Here are the two prokaryotic domains. Let me go back for a second. So bacteria and archaea, those are the two prokaryotic domains. I mentioned them early in the semester. And when we look at them, we move this somewhere. It's not in my way. Not quite sure. Oh, heavens, there's nowhere to put that. Okay. So this says bacteria up here and archaea is down here. For the purposes of public health, we can pretty much ignore archaea because the archaea, these are the extremophiles. They do really well at these ridiculously high temperatures, at like hydrothermal vents at the ocean floors. They do well in ice in the Arctic. They do well at these very low pHs. I mean, just insane environmental conditions that would kill bacteria and would certainly kill humans. And so archaea, there's not a single one of them that causes a disease in humans because they don't generally hang out at 37 degrees centigrade or 98.6 degrees Fahrenheit. The pH, this neutral pH that we have, that's no good for them. So these are not anything that we pay attention to in public health. Also, the parts of the domain bacteria that don't cause a disease, we can safely ignore all of these because you're never going to find any of those on humans. And so for public health, we are concerned with those that can actually live on or in humans and can actually cause disease in humans. And so especially the gram-positive bacteria, that's these over here, and the gram-negative bacteria, and that's this big old group over here. So although the gram-positive and gram-negative are named that because originally it was all based on, what color does it turn in lab, it turns out that those separations were real genetic separations. These groups are actually, it tells you something about their evolutionary history. What's another thing I wanted to point out about this? Oh, I don't know how important this is going to be for you, but if you see something that says low GNC gram-positive, what that means is in the genetic sequence of those ribosomal RNAs, these are ones that have less guanines and cytosine and more adenine and uracil. And it's just a way of kind of separating the groups out a little bit more. It's not super important for our purposes, but that's what that refers to is these have less guanine and cytosine, and these have more guanine and cytosine. And that, I think, is the end of part 1. So I'll see you over in part 2, where we'll cover viruses and prions. See you over there.